All right, so gene size. Before we discuss the structure of genes, we must get a sense of how long genes really are. When we discuss the structure of DNA, transcription, translation, etc., genes were illustrated as if they were only 20 nucleotides long. This is a gross misrepresentation. However, these illustrated genes were abbreviated for the sake of simplicity and the conveyance of basic concepts. So for now on, we will be confronted with visualizing genes within the context of their true lengths. All right, so now let's look at this. Typical lengths of a gene strands are several thousand to hundreds of thousands of nucleotides long. For abbreviation, genes are measured in terms of kilobase pairs. 1 kilobase pair is 1,000 complementarily base paired nitrogenous bases. So in other words, if you compare DNA to a railroad track, as we did metaphorically in lectures previously, 1 kilobase pair equals 1,000 railroad ties. All right, so thus far has been estimated the human genome contains approximately 25,000 genes. So recall that a gene is a sequence of DNA nucleotides in the genome that is required for the production of a functional product. The functional product can either be a protein or an RNA molecule. All right. The great majority of genes, however, code for proteins. However, there are far more than 25,000 proteins in the human proteome. By the way, a proteome is the complete set of proteins encoded by the human genome. The genetic information encoded within the 25,000 genes of the human genome is expanded to encompass the coding of many more proteins via the presence of alternative coding segments within individual genes. In other words, the approximately 25,000 genes of the human genome can encode several different or alternate protein products. In addition, post-translational biochemical modification of proteins significantly increases the variety of different proteins that can be generated from the genes of the human genome. So as a result of alternative coding segments and biochemical modifications of proteins, a repertoire of about 25,000 genes results in the production of over 1 million unique proteins of the human proteome. So here is another figure designed to emphasize the expanded array of proteins that can be synthesized from a relatively smaller palette of genes. So each of these blue boxes here represents a gene along a chromosome, and you can tell that where the different chromosomes are by these dotted interruptions in the DNA molecule. Okay, so let's show some of these genes giving rise to a particular protein, specific protein to that gene within the proteome. Okay, now here you can see that some of the genes of the genome give rise to multiple protein products of the proteome. All right, so in addition, you can visualize the various different forms of the protein created by, or the various forms of these individual proteins here created by post-translational processing. So this is evident from the different multicolored attachments added to some of the proteins. In other specific cases, you may notice that some proteins have been cut or cleaved in different places. This is also a result of post-translational processing. So let's lay the foundation of this same figure. But instead of talking about multiple protein products of genes, we're going to be talking about the multiple functions of those protein products of genes and how those proteins are interrelated in their functions to bring about much more complex functions, okay, that can be observed as being a property or trait or phenotype of the organism. All right. So just as genes rarely have solitary purposes, the approximately 1 million proteins of the proteome do not function in a solitary fashion. Instead, proteins of the proteome work in conjunction with one another to form the diversity and richness of complexity observed on the cellular level. So here we can see the different protein products of the proteome, and here we can see how the different protein products interact with one another, okay? And we call this interaction among the different proteins of the proteome the gene network, okay? And we can see how these interactions then combine to serve within the context of many different specialized cell functions. And your readings that I have that are in coordination with this PowerPoint go into the specifics of different proteins that interact with one another to bring about these complex cellular functions. All right, and I'm not going to go into that right now, but the big picture is that we want you to, over time, hopefully be able to become familiar with the terminology that I've used, all right, as you start to fill in the blanks, okay, fill in the specifics that are within the scaffolding of your emerging knowledge about the basic biosciences. But for the purpose of this class, we're not going to go into the specifics of any of these different interactions of the gene network that I've illustrated. All right. So this rich milieu of protein interaction within and among the trillions of cells of the human body work together to shape complex phenotypes. And a phenotype, again, is the observable properties of a cell or organism. Okay, these phenotypes encompass the distinct and varied physiological, anatomical, and behavioral manifestations that are exhibited in the human being as a whole. Okay, so often genes are presented as being exclusively responsible for a single specific phenotype. However, again, this conception of a gene is far too simplistic. By examining this model of gene expression, it should be evident that many genes not only have multiple products, but that through multiple interactions between their products, aka the gene network, they also have many different effects on various traits, aka phenotypes. Okay? As a result of defects in genes and their products, you can have far-reaching and diverse effects on anatomy, physiology, and behavior, which certainly should be evident in the different diseases that you've chosen to investigate further. So let's go back once more to our original working definition of a gene. A gene is a sequence of DNA nucleotides in the genome that is required for the production of a functional product. And we stated before that this functional product can either be a protein or a functional RNA molecule, such as tRNA. For now, however, we will proceed to discuss gene expression with regard to the production of proteins. All right? So recall that we primarily focused on the central dogma of molecular biology in the previous lecture on gene expression. This understanding of gene expression entails synthesis of a messenger RNA transcript that is complementary to the template strand of a gene. Okay? By complementary, we mean that the mRNA nucleotides are aligned with the nucleotides of the template strand according to the complementary base pairings of the respective nitrogenous bases. Also recall that within the double-stranded DNA molecule, the coding strand is complementary to the template strand. All right. As a result, the mRNA transcript generated from the template strand is the same sequence as the nucleotides of that of the coding strand. The one exception to this assertion is, of course, that the nitrogenous base thymine in the coding strand of DNA is manifested as the nitrogenous base uracil in the corresponding mRNA. Okay, as we discussed several times in previous lectures, this is because the nucleotides of RNA possess a nitrogenous base uracil in lieu of the nitrogenous base thymine. Okay. So as we discussed in previous gene expression lectures, the sequence of nucleotides within the coding strand of the gene are the ones that correspond with the sequence of nucleotides in the initially transcribed messenger RNA transcript that is generally referred to as the coding sequence. Okay. Recall that in the central dogma of molecular biology, okay, uh, which also entails the translation of the sequence of nucleotides of a messenger RNA transcript into a sequence of specific amino acids, okay, that correspond with the sequence of nucleotides within the mRNA transcript, okay. Within this process of translation, sequential groupings of 3 consecutive nucleotides of mRNA called codons are matched with their corresponding anticodons of the tRNA molecules. The anticodons of the tRNA are simply 3 consecutive RNA nucleotides at one end of the tRNA molecule. The anticodons of the tRNA are specifically matched with their corresponding codons on the mRNA via complementary base pairing among the nitrogenous bases of the nucleotides. At opposite ends of the tRNA molecules are amino acids that specifically correspond with the anticodons. At the ribosomes, the tRNAs corresponding to adjacent pairs of codons on the mRNA are aligned, and peptide bonds are synthesized between their respective amino acids. This process is reiterated over and over again along the entire mRNA transcript. As a result, a long chain of connected amino acids or peptides are constructed so that the sequence of amino acids specifically corresponds to the codons along the mRNA transcript. Okay, so remember, again, this is a brief overview of transcription and translation. I know it seems like a lot. It's a lot of jargon. You really need to slow down and pull it apart piece by piece for it to really resonate. And hopefully that's already been accomplished. But if not, please go back to the first lecture in gene expression to go over those different processes. They're no more complex there than I've just briefly reiterated. So let's move on here with the point of this figure. It's important to realize, however, that the initial pre-mRNA molecule synthesized during transcription is not identical to the initial pre-mRNA molecule synthesized during transcription is not identical to the mature mRNA molecule used to synthesize a protein during translation. Instead, larger portions of the initial pre-mRNA molecule are edited out. Okay? The remaining mRNA segments that are reattached to synthesize the final mature mRNA transcript, okay, are actually taken out through a process, okay, by which specific segments of the mRNA molecule are removed while remaining segments are reattached. All right, and we call this process splicing. Okay, and we're going to detail the specifics of RNA splicing shortly. All right. For now, it is important that you realize that segments are edited out of the initial pre-mRNA transcript and that the remaining mRNA segments are fused together to form a mature mRNA transcript. The sequence of nucleotides on these fused segments of the mRNA can be traced back to their corresponding nucleotides on the coding sequence of the coding strand of the actual gene. All right, because that's where all the information came from originally. So the region of the coding strand of a gene that corresponds with the segments of the initial pre-mRNA that are fused together after certain segments are edited out are called exons. And here, within the context of the entire coding sequence of the gene, okay, this part of the gene, okay, which from here all the way to here, okay, which contains the information which inevitably will find itself in the mature mRNA molecule, which will ultimately directly code for the sequence of amino acids in the protein. Okay, these segments here highlighted in blue are known as exons. Okay. These are the segments within the coding sequence that actually contain the information that dictate the sequence of nucleotides in the mature mRNA transcript. So the regions of the coding strand of a gene that correspond with the segments of the initial pre-mRNA that line between the exons and end up being edited out of this initial pre-mRNA transcript are known as introns. Okay, so these white regions of the gene between the exons, and then there's 3 of them here, are known as introns. Okay, you can see how the lines got narrower here as a way to designate that these gap regions between the exons as introns. Okay, you can think about the in and intron as corresponding with intervening, as in intervening sequences. So now we still have some more regions of this gene to cover. So the region of the coding strand of the gene that corresponds with the segment of the initial pre-mRNA molecule that stretches from its beginning to the first exon is called the five prime untranslated region. All right. Understandably, this region is called an untranslated region because it is not translated. It is not translated into a sequence of amino acids. There's no information, even though it's part of the coding strand, and even and it makes it into the mRNA, it does not code for amino acids of the protein that will result inevitably. So the untranslated region at this side of the gene is called the 5' UTR because it's located at the 5' end of the gene. Okay, the region of the coding strand, however, that corresponds with the segment of the original mRNA that stretches from its end, okay, to the end of the last exon is called the 3' untranslated region, or 3' UTR. The untranslated region at this end of the gene is called the 3' UTR because it's located at the 3' end of the coding strand of the gene. So notice that the initiator codon, okay, is marked at the junction between the 5' UTR and the first exon, okay? Because the initiator codon, remember, and this is the part of the gene that corresponds with where the codon will be on the messenger RNA. Of course, this is the place where translation will be initiated on the resulting mRNA molecule. All right. This is where we will start keying together the specific sequence of codons on the messenger RNA, which is also represented and manifested as the triplets of the coding strand of the DNA here that's highlighted in blue as an exon. All right, that's where it's all going to start beginning. Okay. So recall from our previous lecture that in gene expression, the initiator codon is the first codon of a mature mRNA transcript that attracts the first anticodon of the tRNA, and then attached to the tRNA is an amino acid that will become the first amino acid residue of a translated protein. The initiator codon marked on this diagram is the triplet code on the coding strand on the gene that corresponds with the initiator codon on the mature mRNA transcript. Okay, I'll say that again. The initiator codon marked on this diagram is the triplet code on the coding strand of the gene, which corresponds with the initiator codon or start codon, which will manifest itself on the mature mRNA transcript when it's made. Also notice that the termination codon is marked at the boundary of the last exon in the 3' UTR. So that recall from our previous lecture that gene expression, our previous lecture on gene expression, that is, that the termination codon is the last codon of the mature mRNA transcript that is responsible for ending the process of translation. So the termination codon marked on this diagram is the nucleotide triplet on the coding strand of a gene that corresponds with the termination codon on the mature mRNA transcript. Okay, the termination codon marked or stop codon marked on the diagram is the nucleotide triplet on the coding strand of a gene that corresponds with the termination codon on the mature mRNA transcript. So it should be apparent by now that the entire sequence of nucleotides of a coding strand of a gene is not directly used as information for constructing the sequence of amino acids of a protein. There's a lot of stuff here that never actually gets used to directly string together amino acids in a specific sequence, right? Okay, so we see now that the nucleotide sequences that actually code for the sequence of amino acids, that being the exons in a protein, are flanked on either side by 5' and 3' untranslated regions, or UTRs, and are interrupted by these gap regions called introns. In addition to that, even further upstream, and when we say upstream, we mean towards the 5' uh, side of the coding strand of the gene. Okay. In addition, further upstream from the 5' UTR is the promoter region. If we recall from the first lecture on gene expression, the promoter region contains sequences of nucleotides necessary for the initiation of transcription. Further, there's a stretch of DNA downstream from the 3' UTR that can also play a role in the function of the gene. Okay. I'm sorry, got a little ahead of myself there. So what is the purpose of these non-translated regions within and beyond the sequences that code for amino acids of a protein product? So contained within the promoter, introns, and 5' and 3' ends of the genes are the gene regulatory elements. So within the promoter, within the UTRs, and within the introns, you have these special regions called gene regulatory elements, and they are involved for or in controlling the expression of the gene. So these regions are useful. They are there for a reason. So let's look at hemoglobin. So now that we've examined the structure of a gene in closer detail, we will now do the same for the processes of transcription and translation. Following the example in the textbook, we will examine transcription and translation in the context of the beta globin gene. So quick note on beta globin. So if you recall from anatomy and physiology, beta globin is a protein subunit of which there are 2 within the 4 protein subunit adult hemoglobin molecule. The other 2 protein subunits in adult hemoglobin are the alpha globin. So right here you can see 2 alpha globin subunits, and then here are 2 beta globin subunits, beta 1 and beta 2. And then within each of these subunits you have the heme group, okay? And that's the group that actually binds to the oxygen molecule, okay? Takes up oxygen or releases oxygen. So now let's look at this beta globin gene. So the beta globin gene possesses information for triplet codes rather that are necessary for codons which code for 146 amino acid residues. The beta globin gene is located on the short arm of chromosome 11. It is approximately 1.6 kilobases or 1,600 base pairs long. All right. So here we have the short arm of chromosome 11. We can take that out to look at a number of different genes that are within the region of our beta globin, and then look at this very specific region right here in which we have beta globin located here, and we have a variety of other hemoglobin subunit genes that code for other more specific subunits of hemoglobin, which are expressed at different times. All right, the beta globin-like genes. Notice they're all together in the same little region here. Okay. So now the beta globin gene is on the 5' to 3' strand, or I'm sorry, rather, obviously the gene has both strands to it. Its coding strand rather is the 5' to 3' strand. Okay, whereas the 3' to 5' strand serves as the template strand. So interestingly, um, uh, in some genes it may be the reverse, but I just want to give you a heads out here that in beta globin, okay, um, the coding strand of the gene is going to be the 5' to 3' direction. All right. So now I've taken beta globin and I've actually arranged it in a way that is opposite to the way it is arranged in that previous diagram, so that the 5' is over here to the left and the 3' is over here to the right, just because it's easier to keep it in track. Or when I say that, or rather I'm specifying the coding strand, so that the coding strand's 5' is here on the left side, and the coding strand's 3' is on the right side. All right. So within the promoter, upstream from the 5' UTR, I'm sorry, we got a little ahead of ourselves there. Oh, no, I'm where I'm supposed to be. Okay, so let's just hold on and look at this diagram before moving on. So within the promoter upstream, which is upstream from the 5' UTR, there are regions or elements that interact with transcription factors. Okay, and transcription factors are proteins that interact with the gene in such a way that they control the degree to which the gene is expressed. Okay, and among these elements, okay, are the cat box and the tata box. And the cat box is actually the nucleotide sequence CCAAT or CCAAT. It's involved in promoting the transcription of the gene, and it's interesting that absence of the cat box is shown to significantly diminish the expression of the beta globin gene. The Tata box is actually the nucleotide sequence ATAAA. It is involved in controlling where transcription of the gene begins. Transcription of the gene begins about 25 to 30 nucleotides downstream or more towards the 3' end of the gene from the Tata box. So in addition to the cat and tata boxes of the promoter region being right here again, um, uh, there are gene regulatory elements called enhancers. And just like the promoter, the enhancers serve to promote transcription of the gene. Okay? However, unlike the promoter, these enhancers are located in multiple different locations within or in either side of the gene. So in other words, enhancers can be located throughout the beta globin gene, introns, and within the regions beyond the promoters, introns, and extrons of the gene. Okay? It should be noted that these enhancers, such as those that operate in conjunction with the beta globin gene, are associated with genes that code for specific proteins relegated to specific cells and tissues. So when the right transcription factors are produced in specific cells and tissues, these enhancers are specifically activated to upgrade the production of the gene's protein product. So again, just to boil this all down, promoter has very specific elements, okay, which, for example, the cat box and the tata box, okay, for controlling the amount and where transcription occurs. And then you also have other special gene regulatory elements called enhancers, which can be located within the promoter, beyond the promoter, within the introns, within the UTR, all over the place. I mean, so that those are just two different categories of gene regulatory elements that we want you to become acquainted with, okay? Those within the promoter, and then the enhancers, which can be within and beyond those within the promoter. And actually, I just misspoke. I just said they can be within the promoter. The promoter is the promoter. But enhancers can be basically anywhere else outside of the exons, within and outside of the gene. All right. So now we're going to go look at an even, or one more, a third type of gene regulatory element. And this is much further upstream, or more towards the 5' direction of the coding strand of the gene. All right. So remember, as I just pointed out to you, there are many different types of hemoglobin proteins that are expressed in red blood cells or erythrocytes during different times in human development. Many of the genes that code for these different kinds of hemoglobin proteins are associated with a type of gene regulatory element known as the locus control region, or LCR. As the name implies, the locus control region is a kind of supergene regulatory element that paves the way for controlling an entire region of hemoglobin genes. So when this LCR is activated by transcription factors, this entire region of the chromatin, okay, where the genes are located, becomes increasingly suitable for transcription. And this is what the textbook is referring to when it describes the LCR as establishing the proper chromatin context. So why are we viewing all of these gene regulatory elements? While it may be interesting from a scientific perspective, what is relevant from a clinical standpoint is that if damage is done to any of these aforementioned gene regulatory elements, whether they be promoters or enhancers or LCRs, locus control regions, the expression of their respective genes or genes can be diminished or inhibited. As a result, irregularities in the sequence via mutations can be the root cause of many genetic diseases. As many of you research the mechanisms of your genetic diseases, you may find that your disease is the result of mutations that occur in these regions. All right. So back to beta globin. As we just stated, transcription of the beta globin gene begins roughly 25 to 30 base pairs downstream, or more towards the 3' end of the coding strand from the Tata box. Okay. Transcription begins with the transcription of the 5' untranslated region, or 5' UTR. All right. So let's start with that. Let's start showing that. So here we have the non-transcribed strand, aka the template strand, and the transcribed strand. Okay, and now we're going to separate them. We got the exons. For you here, the ones that will actually end up making it to the mature mRNA, okay, and contain the actual triplets, which will code for the codons of the mature mRNA, which will then correspond with a specific sequence of amino acids within the protein. And so here, let's start the transcription process here. And the two strands of the DNA split from one another. All right, and now we see the messenger RNA, or not messenger, yeah, the messenger RNA, sorry, messenger RNA molecule, okay, being constructed along the template strand right here. Okay, so I'm sorry, this is the template strand, and then this is the coding strand. Okay, and here we have the exons within the coding strand right here. Everybody, so this is the coding strand. This is the template strand. We use the template strand to make the messenger RNA molecule, because the template strand is the complementary sequence of the coding strand, and that messenger RNA is the complementary sequence of the template strand. The sequence of nitrogenous bases of the messenger RNA are actually identical to that of the coding strand, except for the fact that instead of thymine, you have uracil, because in RNA, you always have uracil instead of thymine. All right. So again, transcription progresses continuously through the alternating exons and introns and through the 3' untranslated region or 3' UTR. Okay, and again, as you can see here, beta globin has 3 exons interrupted by 2 exons. Okay. So now what we have here is our messenger RNA transcript. This is the pre-mRNA, okay, which includes information that was gained through the transcription of the introns. Okay, so you can see the introns are included, or that the complementary base pairs of, or I'm sorry, the corresponding messenger RNA with the introns within the coding strand of the gene are included in this initial pre-mRNA transcript here. Okay. So transcription then ends at a yet undetermined means at the 3 or at the end of the 3' untranslated region or 3' UTR. Okay, and again, the resulting from this transcription is the primary RNA transcript or pre-mRNA. The pre-mRNA transcript contains a 5' UTR, 3 exons interrupted by 2 introns, and a 3' UTR. Again, all of these components or regions within the primary RNA transcript are all connected and continuous with one another. Following its synthesis, the pre-mRNA transcript detaches from the template strand of the gene, but stays within the nucleus. And by the way, this big smiley face here, this represents the nucleus. All right, so you can see that this original pre-mRNA transcript stays within the nucleus. Okay, so it's at this point that the special process of RNA splicing commences or begins. RNA splicing is the process by which introns of the pre-mRNA transcript are precisely cut out. So while the word splice may sound unfamiliar, think about it this way. Splicing is the process by which specific introns are sliced out of the pre-mRNA transcript. When you merge the word specific, referring to the intron, and the word slice, referring to the cutting out of the introns, you have the word splice. Okay, it's a combination of specific and slice. It's a specific slice. Specific enzymes are responsible for splicing out these introns in specific places. These enzymes cut the DNA according to specific pairs of nucleotides or dinucleotides because there's 2 in a row that these enzymes are looking for that are along the pre-mRNA transcript. Okay. So the dinucleotide, or 2 nucleotides in a row, that marks the 5' cut point of the intron is AG. And this cut is made immediately after the AG dinucleotide. This AG dinucleotide within the primary RNA transcript corresponds with the dinucleotide AG on the coding strand of the gene. Then the dinucleotide that marks the 3' cut points of the intron is GU. And the cut is made immediately before the GU dinucleotide. This GU dinucleotide within the pre-mRNA transcript corresponds with a GT nucleotide on the coding strand of the gene. As the 2 introns are spliced out of the pre-mRNA transcript, the exons are then fused together. All right. So recall that the exons themselves are stretches of DNA that actually code for the specific sequence of amino acids that will compose the protein. As such, also recall that the exons are segmented into nucleotide triplets known as codons. Each specific codon corresponds with a specific amino acid. In the case of many genes, the exons all contain complete sets of codons, meaning that they all possess 3 nucleotides. All the codons have 3 nucleotides. Okay, as is the definition of a codon. In other words, there are complete 3 nucleotide codons adjacent to all of the introns. Okay, so there's a complete, at the junction between the introns and the extrons, you have full, complete um, codons. You don't end up clipping off the codons short when you do the splicing process. Okay, it's a nice clean cut with the ends of the exons possessing nice full codons. Okay, however, in other genes such as beta globin, it may be the case that the exons do not possess complete codons adjacent to the introns. Okay, so the codons flanking the exons have less than the required 3 nucleotides. In this case, the fusion of 2 adjacent exons during splicing completes those incomplete codons. In other words, a specific codon does not exist until after splicing. Specifically, in the case of beta globin, a codon amongst the first 2 exons is split or interrupted by the first intron. This codon is completed only when the first 2 exons are joined together. So why have we considered splicing in such depth? Well, it's important to know about the specific dinucleotides because mutations that destroy these specific dinucleotides impair the proper removal of introns. As a result, the mature RNA transcript that will deliver incorrect information for making a specific protein will now possess new codons within the persistently included introns, okay, which were never intended to be translated by the tRNAs at the ribosome. So the result will be a grossly deformed and dysfunctional protein product. As a result, such mutations result in a variety of genetic diseases. As many of you research the genetic mechanisms of your diseases, you may find that your disease results from mutations that impact such splice sites. Here we see part of the coding strand of the beta globin gene blown up so that we can see all of the individual nucleotides of its code. Okay, you will notice the cat box and tata box highlighted in dark blue and the 3 exons highlighted in light blue. The stretch of non-highlighted nucleotide sequence from the 5' end to the beginning of the first exon is the 5' UTR. The stretch of non-highlighted nucleotide sequence from the end of the 3rd exon to the 3' end of the strand is the 3' UTR. The non-highlighted nucleotide sequences between the 3 exons are the 2 introns. Specific enzymes are responsible for splicing out the corresponding introns in the pre-RNA transcript of the gene. Okay? And remember, these are the corresponding introns, okay, that are located within the pre-RNA transcript. It's the pre-RNA transcript that gets cut, not the gene. So these enzymes cut the RNA according to the specific pairs of nucleotides or dinucleotides along the primary RNA transcript. The dinucleotide that marks the 5' cut point of the corresponding intron of the corresponding pre-mRNA transcript is AG. The cut on the corresponding pre-mRNA transcript will be made immediately after the dinucleotide corresponding with AG, okay, which will also be AG on the pre-mRNA molecule. Again, this AG dinucleotide within the coding strand of the gene corresponds with the dinucleotide AG on the pre-mRNA transcript of the gene. The dinucleotide that marks the 3' cut points of the corresponding intron of the corresponding pre-mRNA transcript is GT. The cut on the corresponding pre-mRNA transcript will be made immediately before the dinucleotide corresponding with GT, and in the pre-mRNA molecule, this will manifest in GU, not GT, because RNA has U instead of T. Okay, again, this GT dinucleotide within the coding strand of the gene corresponds with the GU dinucleotide on the pre-mRNA transcript of the gene. As the 2 introns are spliced out of the pre-mRNA transcript, the exons of the pre-mRNA are fused together. Recall that the exons themselves are the stretches of DNA that actually code for the specific sequences of amino acids that will compose the protein. I know I've said this over and over and over and over again, but there's no harm in repetition. So as such, we call the exons of the coding strand of the gene are segmented into nucleotide triplets, and that each specific triplet corresponds with the codons of the mRNA transcript. In turn, each specific codon of the mRNA transcript corresponds with a specific amino acid. So in this diagram, you can see that the triplets of the coding strand of the DNA aligned with their respective amino acids, which are placed above. You can tell what amino acids they are by the three, by their abbreviations. Okay, so like ARG is arginine, F or PHE is uh, um, phenolphthalein, and et cetera, et cetera, et cetera. Okay. In the cases of many genes, the exons are all complete sets of codons. They all possess 3 nucleotides. In other words, there are complete codons adjacent to all the introns. However, in genes like beta globin, it may be the case that the exons do not possess they do possess incomplete codons adjacent to the introns. In this case, the fusion of 2 adjacent exons during splicing completes those incomplete codons. In other words, a specific codon does not exist until after splicing. Specifically, in the case of beta globin, a codon amongst the first 2 exons is split or interrupted by the first intron. This codon is then completed only when the first 2 exons are joined together through splicing. Okay. So again, why have we considered the splicing in such depth? Well, why is it important to know about the specific dinucleotides that inform the specific splicing of introns? Mutations that destroy these specific dinucleotides impair the proper removal of introns. As a result, the RNA transcript will deliver the incorrect information for making a specific protein. The RNA transcript will now possess new codons within the persistent introns, which were never intended to be translated by the tRNAs at the ribosome. The result will be a grossly deformed and dysfunctional protein product. As a result, such mutations can result in a variety of genetic diseases. As many of you research the genetic mechanisms of your diseases, you may find that your diseases result from mutations that impact such splice sites. About a third of all genes in the human genome employ a strategy of alternative splicing. Alternative splicing is the result of the use of different alternate splice sites in the process of editing out introns and fusing exons of the pre-mRNA. The splicing out of different introns will result in the presence of different fused exons in the mature mRNA transcript. All right. So here we have a gene, okay, and we have these different exons represented by these different shades of blue and gray. All right, and all this information here, including the introns between the exons, will be manifest in the messenger RNA, the pre-mRNA molecule, okay? But if we are only to include 3 of these possible 4 exons within our protein product, we have different choices about how we may wish to combine these exons to form our mature mRNA. Okay, so through alternative splicing, we can end up with 2 different mature mRNA molecules, okay, which will give 2 different proteins, protein A and protein B. Why is this? Well, remember that the codons that will ultimately determine the sequence of the amino acid residues in the protein are located on the fused exons. So by changing the exons that manifest in the mature mRNA product, you will result in different proteins that possess different sequences of amino acids. So while approximately 1/3 of the 25,000 genes in the human genome exhibit alternative splicing pathways, there's an average of 2 to 3 alternative splicing pathways for each of the 25,000 genes of the genome. This is because larger genes that possess multiple splice sites among many exons can exhibit exponentially larger numbers of different mature mRNAs as a result of alternative splicing. So there's a few genes in there that are huge, and they just have a whole lot of alternative splicing going on, which kind of skews the statistics to some degree. So your textbook gives an example of a potassium channel gene. This gene possesses 35 exons with 8 alternative splicing patterns. This can result in over 500 different mature RNA transcripts, each of which would be responsible for informing the synthesis of a unique and different protein. Slightly different potassium channels, in other words, okay, which may be in a variety of different tissues at different times. All right, we're almost getting there. Actually, at this point, I think we're just going to take a quick little break, and then we'll continue on with a little bit more in-depth look at translation, and then that will be it. I'll see you soon.